So, good day everyone. I am Rosemary C. de Guzman and I am the first to discuss Legionella chapter 34. So, Legionella belongs to the family Legionellidaceae, which includes a single genus. Legionella comprises approximately 58 species and 3 subspecies. Legionella pneumophila is the causative agent for Legionnaire's disease, a febrile and pneumonic illness with numerous clinical presentations. So, Legionella was discovered in 1976 by scientists at the Centers for Disease Control and Prevention, so as we called CDC in short, who were investigating an epidemic of pneumonia among Pennsylvania State American Legion members attending a convention in Philadelphia. So, there is a retrospective serologic evidence of Legionella infection far back 1947. So, bacteria resembling Legionella that are capable of living in amoeba have been designated as LLAP or called as Legionella-like amoeba pathogens. So, epidemiology of Legionella. So, Legionella are ubiquitous and widely distributed in the environment. As a result, most individuals are exposed to Legionella species. So, however, few develop symptoms only. In nature, Legionella are found primarily in aquatic habitats and thrive at warmer temperatures. So, kaning nga bacteria, mas capable sila surviving sa extreme range of environmental conditions for long periods. So, study have shown that Legionella pneumophilia can survive for up to 14 months sa tubig with only a slight loss in viability. So, Legionella species have been isolated from most natural water sources investigated, including lakes, rivers, and marine waters, also or as well as moist soil. So, organisms are also widely distributed in man-made facilities, like air conditioning ducts, o kanang cooling towers, potable water, large warm water plumbing systems, humidifiers, whirlpools, and technical medical equipment in hospitals. So, Legionella infections are acquired exclusively from environmental sources. So, wala siya person to person or dili same sa kwan nga makakuha ka aning bacteria sa imong kastorya or sa laing tao. Kay wala pa pud gid siya kanang wala pa gid siya na document nga ingana nga case nga person to person. So, kani siya nga infection is most makuha sa kanang inhalation and aspiration of infectious aerosols are considered the primary means of transmission. So, mo na siya. Iyang primary transmission kay inhalation ug aspiration sa infectious aerosols. So, ang exposure sa aerosols, mas ma-occur na siya or mas makuha niyo mo sa workplace or sa industrial or healthcare settings. For example, kay like nebulizers nga filled with tap water and showers nga have been implicated. So, mostly, dara po nimo siya makuha. Amo na siya example, nebulizer nga na-fill og tap water o ang shower nga na-implicate. So, next one is pathogenesis and spectrum of disease. So, the virulence mechanisms of Legionella species are an important factor in the ability to infect and subsequently multiply within amoeba. So like Hartmanella, Acanthamoeba, and Negleria species. Like that Tetrahymena species, acylated protozoa, and certain host cells. So the organism can also multiply within biofilms, well-organized microcolonies of bacteria usually enclosed in polymer that are separated by water channels that remove waste and deliver nutrients. So, na siya two morphologically distinct forms in HeLa cells. So, first is a highly differentiated cyst-like form that is highly infectious, metabolically dormant, and resistant to antibiotics and detergent-mediated lysis. So, kani siya kay resistant siya sa antibiotics. Second is a replicative intracellular form that is ultra-structurally similar to agar-grown bacteria. So, so, the existence of the cyst-like form may account for the ability of Legionella pneumophilia to survive for long periods between hosts, like amoeba or humans.
Good day, everyone. I am Shilua Mipulay again. In this chapter, I will be covering the life cycle of Legionella in both protozoa and human macrophages. So, binding of microorganisms to receptors on the surface of eukaryotic cells. Second is the penetration of microorganisms into phagocytes. Third is the escape from bactericidal attack, the formation of a replicative vacuole, and the intracellular multiplication and killing of the host cell. So in this figure, this is the Legionella phenomena, cellular trafficking and growth mechanism inside a human macrophage. So first is the sequestering of Legionella within macrophages. Also makes it difficult to treat the infection effectively with antimicrobial agents. As a result, a capable mediated cell Legionella infections require a strong immune response to recover. So, Legionella phenomena has a DOT or ICM secretion system. So, this system possesses genes that are able to trick eukaryotic cells into transporting them to the endoplasmic reticulum. So, Legionella phenomena has 27 genes and is classified as a type 4 secretion system. So, the bacterial secretion systems of type 4 are devices that release proteins and other macromolecules across and into cells. So, Legionella phenomena residing in a membrane-bound vacuole is surrounded by a ribosome-studded membrane derived from the host cells endoplasmic reticulum and mitochondria. So the type 2 secretion system carries numerous genes for enzymatic degradation, including lipases, proteinases, and a number of novel proteins. So, what is legionellosis? Legionellosis is a generic term describing the pneumonic and non-pneumonic forms of infection with Legionella, a spectrum of clinical presentations ranging from asymptomatic infection to severe life-threatening diseases. So we have three primary clinical manifestations. So legionellosis varies in a severity from a mild febrile illness to a serious and sometimes fatal form of pneumonia and is caused by exposure to Legionella species found in water and potting mixes. So the first of the three clinical manifestation is the moto pneumonia. Then second is the manifestation of Pontiac fever or the mild flu-like illness. It is a non-fatal influenza-like infection. And the third is the rare extrapulmonary sites such as wound abscesses, encephalitis, and endocarditis. So this is the three primary clinical manifestation: the pneumonia, Pontiac fever, and extrapulmonary.
Hello, good day everyone. My name is Maryphil Yvonne Gonzalez Zafra. I'm gonna tell you about the laboratory diagnosis of Legionella. The specimen collection and transport. Patients with Legionella disease usually have detectable numbers of organisms in their respiratory secretions even for some time after antibiotic therapy has been initiated. Pleural fluid has not yielded many positive culture in studies performed in several laboratories, but it may contain organisms. The organisms are hardy and are best preserved by maintaining specimen in a small tightly closed container to prevent desiccation and transporting them to the laboratory within 30 minutes of collection. If a longer delay is anticipated, specimens should be refrigerated. If moisture of the specimen cannot be ensured, 1 ml of sterile broth may be added. The specimen processing, all specimens for Legionella culture should be handled and processed in a Class 2 Biologic Safety Cabinet or BSC. Brief treatment of sputum specimen with hydrochloric acid before culture has been shown to enhance the recovery of Legionella. Respiratory secretion may be held up for 48 hours at 5 degrees Celsius before culture. Tissues are homogenized before smears and cultures are performed and clear. Sterile body fluids are centrifuged for 30 minutes at 40,003 grams. Blood for culture of Legionella may be processed with the licensed centrifugation tube system and plated directly to buffered charcoal yeast extra agar or BCYE agar. The specimen collected by bronchialveolar lavage are quite dilute and therefore should be concentrated at least tenfold by centrifugation before culturing. The direct detection method, the stain. The Legionella several species are gram-negative bacteria with a thin cell wall but stain poorly in the gram procedure if neutral red or safranin is used as the counterstain. The antigen, one approach to direct detection of Legionella in clinical specimen, is the direct immunofluorescent antibody or DFA test for respiratory secretion. So the molecular methods, polymerase chain reaction or PCR assays has been extensively used to detect deoxyribonucleic acid or DNA from environmental and clinical samples, particularly those from the respiratory tract. So the cultivation BCYE is also prepared with ACEs buffers or amido and amino ethylene sulfonic acid and the growth supplement system required by Legionella, yeast extract, alpha ketoglutarate, and iron. Specimens obtained from sterile body sites may be plated to media without selective agents and may be also inoculated into special blood culture broth without sodium polyethanol sulfonate or SPS.
Good day everyone, I'm Aurelio C. Martir and I'm going to report these remaining topics. So first topic is approach to identification. So definitive identification requires the facilities of a specialized reference laboratory. Suspect colonies should be gram-stained to determine whether bacteria are small to filamentous gram-negative rods. Clinical laboratories probably perform sufficient service to clinicians by indicating the presence of Legionella spp in a specimen. Serologic typing is a simple method and should provide sufficient characterizations. If further identification is necessary, the isolate should be forwarded to an appropriate reference laboratory. Next is serodiagnosis. Most patients with legionellosis have been diagnosed retrospectively by detection of a fourfold rise in anti-Legionella antibody with an indirect fluorescent antibody test. Diagnostic efficacy associated with serologic testing increases with the collection and testing of acute and convalescent paired sera. So it is essential to correlate serologic findings with the patient's clinical presentation because of the variation in antibody response associated with legionellosis. Next is antimicrobial suspectibility testing and therapy. In vitro suspectibility studies are not predictive of clinical response and should not be performed for individual isolates of Legionella. So clinical response usually follows within 48 hours after introduction of effective therapy. Clinical response usually follows within 48 hours after the introduction of effective therapy. Nabalik na eh. So next is prevention. The effectiveness of other approaches to the prevention of Legionella infections such as the elimination of its presence from cooling towers and potable waters is uncertain. An important piece of information for surveillance of the disease is the history of exposure. The incubation period of Legion- Legionellosis is normally between 2 and 10 days. Many countries have developed guidelines or regulations for the control of Legionella in water systems and for the prevention of Legionellosis. So that may be all. Thank you for listening.